happening. So electron transport, right? Everything, kind of all of this, all these metabolic processes are very reliant on redox reactions, right? Things are losing electrons, things are gaining electrons, just kind of over and over and over again. And so you create these electron transport chains, right? Things are being taken off, put on, and moved around, right? And so in redox reactions, electrons are moved from one atom to another. But like we mentioned in chapter 6, right, this is never super efficient. We always kind of lose a little bit, right? We pull this electron, we gain a bit of energy, but a little of it kind of falls off to the side, right? So this kind of stepwise energy loss in electrons, right? So you've got this bound electron with some set amount of energy, you pull it off and harvest some energy, but you lose a little bit. So the next time you harvest energy with this electron, you're going to lose a little bit, right? Kind of over and over again through this series of redox reactions. And eventually you're going to have this little kind of super tired electron, right? And it's not going to be, honestly, it's just not going to be of much use anymore. And so at the end of these kind of electron transport processes, we end up just giving it to what we call a final electron acceptor, right? We're just like, this electron's done, just stick it to something and be done with it. It is frequently oxygen, right? Which kind of makes sense, right? Because oxygen is highly, remember, oxygen's highly what? It's what makes molecules polar. One of the things that makes molecules polar is that oxygen is very electronegative. Yeah, it's electronegative, right? It's highly electronegative. It's very greedy as far as electrons, which subsequently means it's not very picky. So it doesn't care that this little electron is kind of super tired, kind of not of much use. It's still like oxygen's still going to pick it up, right? So frequently oxygen is our final electron acceptor, right? But not always. So depending on kind of in your body or in an organism, depending on what is done with that tired little electron, right? It's kind of how we name different kinds of respiration. So we, generally speaking, do aerobic respiration, right? Because our final electron acceptor is generally oxygen, right? Because we breathe in, we have oxygen available. There are organisms that live in environments where there's not a lot of oxygen. They don't have oxygen available. So they attach their little tired electrons onto some other inorganic thing. And so it's anaerobic, right? This kind of, right, aerobic has to do with the presence of oxygen. So anaerobic would be without oxygen. And then there are a few organisms, things like yeasts, that will attach their electrons to other organic molecules, which is fermentation, right? And we'll talk about fermentation in more detail kind of at the end of this chapter. But kind of our general assumption and the way we're going to walk through respiration is aerobic respiration, kind of what our bodies are doing most of the time. Unless, and we'll see this example later, when you're doing certain activities, your tissues can become oxygen depleted. And then our bodies can kind of in small spurts do a bit of anaerobic respiration, but we'll get there. That's the end of the chapter. All right. So again, just seeing these electrons, right, getting moved around, this kind of electron transport through this respiration process until we've used up all the energy we can pull from them, and then we just kind of stick them to an oxygen. We're done with them. All right, so this is kind of where we actually left off on Friday. Glucose, right, that 6-carbon sugar, is oxidized to CO2 in a stepwise way. We don't just immediately kind of oxidize the whole molecule, bust it all apart, right? We are kind of piece by piece peeling off these electrons and breaking these bonds in this kind of stepwise way, right? Because if you were just pulling these electrons, these high energy electrons, and putting them immediately onto oxygen, that process is called combustion, right? And your cells would literally, if they were trying to do that, just burst into flames. You would spontaneously combust if you didn't kind of very carefully, right, kind of pick these glucose molecules apart, right? So that's what the process of respiration is going to be. It makes it sound a bit like diffusing a bomb, but kind of, right? Because if you're just breaking bonds by adding oxygen, that's like, I mean, that's like a campfire at that point. So we've got to do this stepwise process where we kind of dismantle and remove, kind of harvest the energy out of these molecules. Now, so since we're having to kind of pick these electrons off and slowly pull the energy out of them, we've got a number of carriers, right? We talked about NAD becoming NADH, right? That's one of our options. And so different electron carriers will kind of have different skills and abilities, I suppose is one way you could think about it. So we've got three main types we'll talk about. And so we've got these soluble carriers, soluble in water, that will just move electrons from one molecule to another, right? They can cross membranes, they can do, they're kind of like, they can kind of do whatever they want, right? Kind of queen and chess of electron carriers. They can just move things around and kind of go wherever they like. We then have membrane-bound carriers that form these redox chains, right? So these membrane-bound carriers that are going to be in a membrane. So they're not going to be carrying anything anywhere. They're just going to be passing things kind of within their space, within their little kind of processing areas. So we've got these membrane-bound carriers that form redox chains, which we'll see in respiration. And then we have other carriers, right? So this would be like NAD that we saw earlier. I mentioned FAD. FAD is going to be in this third, and we'll talk about it later. You don't have to like memorize FAD right now. But we've also got some carriers that aren't soluble, that can't cross membranes, can't kind of go as far. And so they kind of have to, whatever little area they exist in, they can move around. They're not bound to a membrane, but they can't cross one either. So they're kind of stuck in their own way, right? But they're still moving electrons around in their little space. All right, so we've got these three kinds of things that are moving electrons around as we dismantle this molecule, right? And remember, right, so they can all be oxidized and reduced, right? So if we're oxidizing a molecule, the carrier is getting reduced, right? It's receiving the electron. It can then carry the electron somewhere else and be oxidized, where it gives it to something else, right? So we'll see it as we pull glucose apart, we're going to reduce these electron carriers. The electron carriers are going to take those electrons, deliver them somewhere else where they get oxidized and give that electron to something else. All right, that's fine. All right. So NADH, one of those electron carriers, is used to make ATP. So if we have a lot of ATP, we don't really need NADH to make more ATP. And so we can actually, if we start accumulating too much NADH, right, we've got enough ATP, we don't need all this NADH, it can actually be reduced to fatty acid precursors. So this is where you've taken in kind of like too many calories, you've taken in too much energy. And so this would be the part in the process where the energy you're taking in begins to get stored as fat, right? We don't need the stuff we're taking in to make ATP at this point. We have enough. We reduce NADH to kind of the beginnings of these fatty acids, and they're used to build lipids for fat and energy storage to be used later, right? We're not going to toss that energy, right? We might need it later, so we'll store it in the form of lipids. Which again, interestingly, kind of maybe you wouldn't expect, is a process that also relies on the electron carriers. All right, so like we said, harvesting energy, series of redox reactions, right? It's released so we're harvesting it in stages. We also release it in stages. We're doing all of this kind of very stepwise. So we're harvesting all of this energy step by step by step by step, and we're going to pass these to electron carriers in the mitochondria in the electron transport chain, right? And so this is the electron transport chain are bound, they're in a membrane, they're bound in the mitochondrial inner membrane, right? And so we're going to pass the electrons to the electron transport chain and release that energy, again, in a stepwise process. Which we will look at kind of as we walk through respiration proper. So we harvest energy in a series of redox reactions, and then we release energy in a series of reactions within the mitochondria, right? Which makes sense. It's the powerhouse of the cell. It's where the power is being released to do the things generally. All right. ATP again, some more. So ATP, fundamental in metabolism, powering all of these reactions, we know. But how are we getting it? Like, how are we making it? We're making it in basically two ways that we'll worry about anyway. Synthesized by cells in two ways. So we have, they're funky words, we'll explain them. A substrate level phosphorylation and an oxidative phosphorylation. Substrate level phosphorylation, we can unpack it a little bit, right? So phosphorylation is just adding a phosphate. That's all that means, right? Which makes sense because we're probably turning ADP into ATP. So we've got a little molecule with 2 phosphates. We needed to have 3. So we're going to put a phosphate on it. If we're talking about substrates, what are we talking about? What we talked about in chapter 6. What uses substrates? Those proteins that do things. That make reactions go faster are called enzymes, right? Enzymes bind to substrates. So it looks kind of like you would expect. We've got an enzyme, we've got an ADP, right? A used up ATP, and we're going to attach, we'll get to PEP later. But we've got this other little kind of cofactor that brings in a phosphate. And at the substrate level on this enzyme, we're going to attach this phosphate. We're going to phosphorylate ADP, and we make an ATP, right? So we can have substrate level phosphorylation where we use an enzyme to make ATP. Easy enough. We can also have oxidative phosphorylation. And this is where, and we'll see this in the electron transport chain. This is just like little ATP factory. So oxidative phosphorylation is where we synthesize ATP by using the very aptly named enzyme, ATP synthase. And so, but in this case, we're going to use this proton gradient we're going to create using that electron transport chain. We're going to make a proton gradient inside the mitochondria that's going to power this. It's kind of a wacky thing. This weird membrane pump, right? And we're going to make ATP that way. Right, so we've got 2 options. This only happens, oxidative phosphorylation only happens in the mitochondria along the electron transport chain. Substrate level phosphorylation is kind of anywhere else that we might want to be making ATP. All right, and we will see both. All right, so which gets us to main show, aerobic respiration. So we're going to be we're going to see this figure multiple times. So you don't need to it's just here for your reference. So what this diagram shows, so here's our mitochondria right here. And so what this is showing that aerobic respiration, part of it happens kind of out in the cell, specifically the part called glycolysis. And then everything else happens inside the mitochondria, right? So we've got glycolysis that occurs outside the cell, right? And glycolysis produces pyruvate. We're going to oxidize pyruvate. It makes sense. We're just going to oxidize the heck out of this glucose until there's nothing left. So we oxidate pyruvate. We go through the Krebs cycle, break it the rest of the way down. And then we have the electron transport chain where we start generating a heck ton of ATP, right? So broadly, that's all of respiration, but we're going to kind of break it down a little further, a lot further, right? So let's start with glycolysis. We'll start with step 1. We're going to talk about glycolysis, talk about how much energy you actually get from the process of glycolysis. And then we'll talk right here's where we'll compare aerobic and respiration fermentation. Now, Glycolysis, like I said, happens in the cytoplasm. Everything else happens in the mitochondria, right? So glycolysis occurs in the cytoplasm and it splits glucose basically in half, right? Glucose is a 6-carbon molecule. We're going to break one of those bonds, right? Pull it apart and end up with 2 3-carbon molecules. Takes us 10 reactions to get there. You're not gonna have to memorize all 10 reactions, don't worry, right? Net gain for glycolysis, right? And here's kind of the simplified version, right? Glycolysis nets you 2 ATP molecules for every glucose. Not amazing, if I'm honest. There's not a lot of ATP, right? So we're going to split glucose in half, create 2 3-carbon molecules, and we're going to net 2 ATP. We're actually going to create 4, but we're going to have to spend 2. So we only end up with 2 at the end of the day in glycolysis. Now, the way I have these slides set up, real quick, the second version of each of these slides is going to be a much more complicated one. You do not have to know this, right? This is just kind of to help us explain it, right? Your kind of explaining level for me is more this version of the figure, all right? So when we look at these big kind of molecular structure diagrams and things, don't get super stressed out. I'm not going to make you draw carbon rings and stuff on the exam. It's not going to happen, right? It's just kind of for our talking through it purposes, right? So I do better if I've got pictures in front of me personally, right? So we've got 10 total steps. The first 3 lumped together, and I'm fine with lumping them together, are called the priming reactions, right? Because the whole purpose of glycolysis is to split, right? We want to split this carbon ring in half. So there's some things that have to happen to kind of get it ready to break it in half. So for starters, we're going to attach 2 phosphates, right? Which is going to cost us 2 ATP, right? Because that happens, right? When we spend an ATP, the phosphate kind of sticks to it, right? So we're going to spend 2 ATP and end up with this 6-carbon molecule with 2 phosphates stuck on. All right. What does that look like? So kind of long form, right? We've got a glucose, we hit it with an ATP, so now we've got this glucose with a phosphate on its carbon 6. We shuffle it around, you'll see a lot of isomerases. I'm also not going to make you memorize a bunch of enzymes, don't worry about that, right? But you'll see isomerases in here a lot. It's just kind of shuffling and reshaping the molecule, right? So we make this by spending an ATP, we kind of shuffle it around into this kind of 5-carbon ring. And then we spend another ATP to tack that second phosphate on. So end of the day, we've taken our 6-ring carbon and had tacked a phosphate onto either end, right? Basically onto the 1 and 6 carbon, right? Onto either side, which is exactly what that little picture was showing, right? So we've got these priming reactions to get these phosphates on there. And we have cleavage reactions, right? Cleavage is, right, we're going to cleave it in half, right? So reactions 4 and 5 make that happen. We've got this 6-carbon sugar diphosphate, right? It's 6 carbons, it's a sugar, it's got 2 phosphates, right? So we're going to split it into 2 3-carbon monophosphate sugars, right? Those are just, right, all of those biology words that say exactly what we just said, right? It's got 3 carbons, it's got 1 phosphate, it's still a sugar of some form, right? They're not going to come out exactly even. You don't need to remember that one of them is initially a dihydroxyacetone phosphate, right? Basically, ultimately, we're going to isomerize it again and end up with 2 glyceraldehyde 3-phosphates, or G3Ps, right? So we've got a 3-carbon sugar with a phosphate, 3-carbon sugar with a phosphate, okay? Reactions 4 and 5 split the 6-carbon molecule in half. Each half has a phosphate. Again, just to kind of see it, right? This is where we left off. We split it in half, 2 3-carbon sugars with a phosphate, 2 G3Ps for our purposes. Again, don't stress out about those figures too much. It's just so you can kind of see what's happening. Now we're going to finally get a bit of ATP out of here, right? Which was kind of the whole point. So at reaction, this is 6 through 10. Reaction 6, we're going to oxidize, remove an electron from, right? Each of those G3Ps, stick an additional phosphate on, because why not, right? And so you see this happening here, right? When you oxidize it, so we're going to oxidize this G3P, which means we've reduced NAD to NADH, right? Because it's pulled the electron and it's taken a little hydrogen on with it. So it loses its positive charge, gains a hydrogen, heads off to the mitochondria. We'll see him later, right? We also add a phosphate, right? Then we're going to have these substrate level phosphorylation reactions, right? We're going to take this phosphate, we're going to convert it to ATP, right? Same on for this 3-carbon sugar, right? It's happening on both sides, right? So we oxidize G3P, we attach an extra phosphate, and then we pop both of those phosphates on through substrate level phosphorylation and make 2 ATP for each of those G3P molecules, so total 4 ATP. 7 through 10 is kind of a bunch more of those shuffling reactions. We end up with a 3-carbon pyruvate for each one. Okay. So do I keep all that text if I go ahead and All right. We're just going to look at this next slide. Don't like, it's okay. It's okay. Right. Don't, it's fine. Right. Because we have these 2 single phosphate sugars. We're going to follow one of them down. Right. So we oxidize it, send off an NADH, add another phosphate. We create an ATP. We lose 1 phosphate. Shuffle, 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 shuffle. Create another ATP, lose the other phosphate. And we're left with pyruvate. Right. So we oxidize them, we phosphorylate, make ATP, we remove some water, that's fine. We end up with 2 PEP molecules, which we then use. We're left with 2 pyruvates. Okay, we'll leave that behind. Right. So start with these 2 single phosphate sugars, add another phosphate, create 4 ATP. 2 pyruvates. Final count. We spent 2 ATP to kind of, so here's all of the kind of, as we're concerned with it, process together, right? We spent 2 ATP to phosphorylate the sugar, split it in half, we oxidized it and put another phosphate on, we generated 4 ATP, so we've got 2 pyruvates, we made 4 ATP, net yield is 2, right? Because we have to spend 2 in the beginning. So we have to spend 2 to get 4, net 2. All right. So for studying purposes, if you can kind of talk through it at this level, that's sufficient, all right? If you want to look at molecules, if kind of seeing them visually helps you kind of remember what's going on, they're also in the slide for you. If they're just confusing, then you can let them be. It's fine. All right. So the yield is low, but the process is relatively efficient. It's reliable kind of in that way, right? So we break glucose in half, but we only really capture 40% of the energy. It's not like the worst process, but it's honestly not the best. It works, right? So we break glucose in half and we can pull about 40% of that bond energy and make a couple ATP. All right. Right, and so when we start to think about pathway evolution, right, because that was actually like if you pulled apart and like looked at every single one of those little enzymatic crazinesses that were going on, it was a pretty convoluted path. But it's that kind of evolutionary thing where as long as whatever your body is doing is helping you survive, then it kind of sticks around. And so you can end up with these slightly convoluted processes that do enough. Right. Now, additionally, kind of the way we talk about pathway evolution, those G3P reactions, right, at the very end, were the ones that happened first, right? Making G3P from glucose happened later, right? So your body, well, not our bodies, little single cell bodies most likely, was able to break down that G3P kind of from the middle down to make those ATP first. And when it couldn't find G3P, turns out you can break glucose in half. You have to spend a couple ATP to do it, but you can use glucose, right, to kind of facilitate that process. So again, that kind of back to front evolution. All right. So what's left? What do we do next? What do we do next? So we still got pyruvate. It's still a 3-carbon chain. We've got 2 of them, right? We still got 2 3-carbon chains. There's still energy in those bonds. We've accumulated some NADH that's carrying those electrons around. Surely we can do something with that. So what is step next? Let's talk about the NADH first. Where is it headed? Right? Well, we're going to need more NADH, right? Because we filled it up with electrons and protons. But we need more NAD to continue glycolysis. So we've got to have some source of NAD plus, right? So we've got 2 options. Either we're doing aerobic respiration and oxygen is going to handle that electron for us and kind of move it out of the way so NAD plus can go back into glycolysis. So we can have NADH oxidized by oxygen, or if we're lacking oxygen, right, we can do fermentation, where we oxidize NADH with some other organic molecules. Right, so we've got options to get NADH back to NAD. All right. So, pyruvate. Yeah, that's fine. So, right, NADH is going to either have to meet up with oxygen or an organic molecule, or something. Now, if we have oxygen, which is kind of our default form, the fate of pyruvate depends on the NADH. So if we have oxygen, which is us, right, we go into the mitochondria and oxidize pyruvate into acetyl-CoA, right? So oxygen's available. We come into the mitochondria to oxidize our pyruvate. We're going to decarboxylate it, which just means we're going to break those last carbons apart, decarboxylate, right? We're going to, oops, did something just, oh, hold on. Oh, come on, Zoom. I believe in you. It is refusing to let me see the chat. Let's come out real quick. Oh yeah, so pyruvate is, good question. Pyruvate is just that 3-carbon molecule that we were left with after glycolysis. It's like the leftovers of glycolysis are 2 3-carbon molecules. They are called pyruvate. And so if oxygen is available, NADH is going to kind of hang out for a second. The pyruvates are going to move into your mitochondria and get completely broken apart, basically, right? So they're going to get decarboxylated. We're going to remove carbon by carbon by carbon, right? We're going to be left with this little acetyl group. We're going to bond that to coenzyme A. So then we've created acetyl-CoA, which will be used in this laptop. Come on, I believe in you. In the Krebs cycle, all right? So before we leave this, right? So just, we're going over this really quick. Oxidation of pyruvate just kind of happens really quickly, right? So we've got pyruvate, We're going to break off, decarboxylate it, loses CO2. We're going to reduce another NAD to NADH. We're going to attach it to a coenzyme A, which gives us acetyl-CoA, right? So here's pyruvate oxidation. It's just a single reaction. This is kind of where we move into the mitochondria, right? Which gets us to the next step, the Krebs cycle, which is a bit more involved. So the Krebs cycle is where we're going to pull lots of electrons off, honestly, right? And go through a long series, again, of oxidation reactions. They're all kind of long series of oxidation reactions. All right, so we've got Krebs cycle or citric acid cycle. If you've heard this before, sometimes people call it the citric acid cycle. And at this point, we are in the mitochondria, right? We oxidize pyruvate as we come into the mitochondria. We're completely in the mitochondria. We've got 9 reactions. This time we're only going to yield 1 ATP. Honestly, this doesn't sound super impressive as we're going through it, right? But we're going to, through all of these oxidation reactions, we're going to reduce a lot of NADH and FADH2 to send to the electron transport chain, which is where this is all going to become worth it. Okay. So Krebs cycle, or the citric acid cycle. So first step is a condensation reaction. So oxaloacetate is kind of the beginning molecule that's like waiting for us. Here in the mitochondria, there's just an oxaloacetate hanging out, waiting for our little leftover acetyl 2-carbon acetyl-CoA to come in. Coenzyme A is going to kind of like, I don't know, bug off, and these little 2 carbons are going to attach to oxaloacetate. Right, so oxaloacetate is kind of what's carrying these last 2 carbons around to kind of finish breaking this molecule down. Right. This reaction, right, acetyl-CoA coming in and putting these 2 carbons onto oxaloacetate is irreversible. So once we get here, we're committed to the Krebs cycle in this step 1. But when there is a lot of ATP, this reaction is inhibited, right? So this is one of those feedback inhibition situations, right? We're sending things to the electron transport chain to make ATP. When we have enough ATP, ATP creates kind of feedback inhibition with this beginning reaction and stops it here. So we attach our little 2-carbon leftover molecule to this 4-carbon carrier. Isomerization happens again. Don't worry, we're just shuffling it around. So we shuffle it around a bit, make this isocitrate. Now we do some stuff. So in reaction 4, we have oxidative decarboxylation. So we're going to oxidize the molecule and decarboxylate it. Carboxyl, remember from lab, carboxyl is just CO2, right? A carboxyl group. So we're going to oxidize it, yield ourselves 2 electrons, reduce NADH, send it off to the electron transport chain. So it's bopped off to do its thing. And then we decarboxylate it. We remove this carbon. So we yield a CO2, and now we're down to this 5-carbon molecule, which if we remember, this is kind of, I don't know, where you might want to start drawing yourselves pictures, right? The initial 4 carbons here are just a little carrier. It's like a little bus that our last little 2-carbon was on. So we have 1 carbon left from our initial pyruvate, right? So we've got 2 going through here at once. So we got 1 carbon left to break off anyway. So we've got a carbon broke off, 5-carbon molecule left. So we just do it again, right? Oxidative decarboxylation. So we oxidize this carbon molecule, reduce an NAD, cut off a CO2, and we're left with this little 4-carbon carrier, which is kind of like what we started with, right? We're going to add another acetyl-CoA, acetyl coenzyme A, which just kind of like comes in and helps us out when we need it. So coenzyme A comes in. We're going to make an ATP right here. So we've added that little coenzyme A. We're going to break the bond between carbon 4 and the acetyl-CoA and coenzyme A. I don't know why I keep saying that wrong. Right? We're going to release energy, we're going to make something like ATP. It's called GDP. That's fine, right? So we're going to make this GTP, and GTP is going to take its phosphate and put it to ADP, makes an ATP. That sounds ridiculous when you say it out loud. But basically, end of the day, we just make another ATP. That's all we've done, right? We just had this kind of intermediary carrier pull the phosphate off of our little 4-carbon molecule and attach it to an ADP to make ATP. So short story, we use coenzyme A to make one more ATP here. You'll notice that's actually the only ATP that we generate in the citric acid cycle or the Krebs cycle. Right, so we break off this carbon and reduce an NAD. We break off a carbon, reduce an NAD, create an ATP. Now, we're back to this little 4-carbon molecule. If we can get anything else out of here, we probably can, and we will, right? So we're going to oxidize this molecule again, but right, we've oxidized it and shuffled it around and broken stuff off. There is no longer energy sufficient to reduce NADH, right? We've worn this thing out. So now FAD comes in. FAD is a little less picky. It doesn't need as much energy to get reduced. So we can reduce FAD to FADH2. That's cool. So it pulls its electron and its hydrogen off of this molecule. But FADH2 is now stuck inside the mitochondria, which is fine because it's kind of where we need it anyway. So it just kind of heads off to the electron transport chain. Can't go anywhere else, but that's fine. It's kind of where we need it. All right, now we need to remake oxaloacetate so that this cycle can happen again. So reactions 8 and 9 are just to make oxaloacetate again. It's kind of our starting point. So we're going to add some water and then oxidize it, right? So we're going to add some water and then pull an electron off, which will, through a series of kind of things happening that we're not worried about, give us this oxaloacetate again and also reduce one more NADH. We're going to kind of liven the molecule up with a bit of water, oxidize it again, and end up with our starting molecule and one more NADH that heads to the electron transport chain. Right. Now this oxaloacetate is free to join up with another little 2-carbon leftover molecule that's attached to that coenzyme A and do the whole thing again. I have gone over time. Somebody should have thrown a pencil. All right. So we'll stop there. Sorry, I lost track of time. All right, so we will meet up Wednesday and I think should finish out this chapter. Again, feel free to let me know what you're thinking, what you're feeling about